Good morning, good afternoon, good evening to all. Thanks for joining us. Welcome to everyone. A special welcome to our new colleagues from Vietnam, from Kazakhstan in Nur Sultan, from Pakistan in Kotri, and India in Pune, all new to the endTB and endTBQ team since our last in-person meeting. Just want to thank you all again, acknowledge that there is no way that we could do all of this incredible work without you. We want to spend this time sharing with you a little bit of a snapshot of the descriptions of the endTB and endTBQ trial populations, begin to share a little bit of the aggregate study results, nothing that requires unblinding for us. I'll share some observations on what we see and some questions for you guys to think about throughout. At the end of this presentation, I'll also share some prompts for future discussions and questions. You can use these prompts to frame your own conversations in the meeting discussion platform or come up with your own ideas. Anything you'd like to discuss further. So let's start with some great news. Thanks to all, the enrollment is complete in output 2 of the endTB project, also known as the endTB trial. We reached this huge milestone in the middle of October. The last patient last visit is scheduled for or anticipated for April of 2023. And ultimately, the distribution of the study population is really quite remarkable, with about 18% of study participants coming from Africa, 38% from South America, 17% from South Asia, and 26% from Central Asia or Eastern Europe. So really good distribution for representativeness, which is something you'll hear me mention a couple of times throughout the talk. And then you can see, so it's 7 countries ultimately enrolled across 12 sites, 1,539 patients were screened. 754 were randomized. I'm only going to be reporting on 753 in this talk, just because that's what was available in open clinic at the time that the data were locked for this, and more than 17,500 study visits, more than 17,500 study visits have been completed so far in this trial, thanks to you all. Talk a little bit more about the screening in particular. So you can see from this slide that just under half of those screened were actually excluded just under half were randomized, just over half were excluded. Almost 80% of the exclusions were related to factors like either not having confirmed resistance to rifampin in a study lab, inability to detect MTB in either a first or second line molecular test, or quinolone susceptibility not being confirmed. Since more than a third of these were because of quinolone resistant or indeterminate results, that bodes well for endTBQ. There are some negatives about our exclusion criterion that could bode poorly for endTBQ as well, which we'll discuss a little bit further. Just want to note the substantial decline in the proportion of exclusions due to investigator discretion as a reason, and congratulate you all on that and on really focusing on the protocol defined criteria. And then there were notably very few excluded because of prior exposure or resistance to study drugs. And this is something that we'll need to keep an eye on in endTBQ, as time goes on and prior exposure may be more likely. This is the reasons for exclusion by study site. You can see that there's substantial variability with the exclusion rate being much lower, say in Georgia and in KZ3 and also in PE3, and exclusion being much higher in India 2 and in Pakistan 2. Notably, nearly all of the exclusions occur at screening, only about 2.3% due to results that were confirmed at or identified at baseline. The population in the endTB trial cohort remains young, just over 30 years, and they remain majority male. As you're all aware, these are often patients with really complex health and social issues. And just as an example, we have a few measured comorbidities here. You can see that 11% reported diabetes mellitus at baseline. Notably, roughly 100 had abnormal A1Cs at screening or baseline. Later, we'll examine the overlap between the self-reported diabetes and these A1C results. We haven't done that yet, and we'll be able to better classify the prevalence of diabetes in the cohort. 14% had HIV co-infection, and there were lower prevalence of seropositivity for active hepatitis B infection and for hepatitis C. This is just to give you a sense of the variability in these, the three viral diseases that we showed in the last slide. So you can see that HIV was present in more than 60% of the Lesotho participants and nearly 60% of the South Africa participants, with tiny numbers from the other sites. And in contrast, the Hep C, represented by this sort of dark teal, was limited to occurring in the Kazakhstan, Georgia, and Pakistan sites, with one additional case in Lesotho. None of these comorbidities have been identified among any of the endTB trial participants in India to date. This gives you a sense of the prior exposure to tuberculosis. Was reported at 36% in the total population. Again, a lot of variability within and between countries. You can see that the lowest prior TB reported was in Peru, and highs were in South Africa and Pakistan too, respectively. And notably, there's a substantial difference between PK1 in Karachi and PK2 in Kotri. Among the other kind of prior exposure question we're looking at is the proportion who were receiving treatment at the time of screening. And again, this is very variable, with almost no patients or no patients in Peru and Lesotho and India and Georgia on treatment at the time of screening, and much higher percentages in Kazakhstan. This may reflect differences in recruitment strategies and access to peripheral health facilities. So all of the patients are started in the trial inpatient, and they may not actually get considered for the trial until they're actually inpatient and may already be on treatment disproportionately. And I guess it's a question that I would pose to the study teams. Is it possible that in places where such a high proportion is on treatment at screening, that we're missing potentially eligible patients? And I guess conversely, in places where there's a very low proportion on treatment, is this because those receiving treatment are generally getting treatment with the study drugs, bedaquiline, delamanid, linezolid, or clofazimine, and therefore have to be excluded? Might we be able to reach this population sooner, before they start getting routine care with those drugs. Obviously, these questions related to recruitment are really more relevant to Q, or only relevant to Q, and no longer relevant to NTB. This is just a little summary of the status of enrolled patients, which really shows more amazing progress in that among patients who had been randomized by the time this data set was locked, more than half had completed treatment. And of those, 2/3 had completed follow-up, either the full 104 or the hybrid at least 73 weeks. There are another 136 who are still in post-treatment follow-up, and 231 who remain on study treatment. 12 or 3% have discontinued follow-up earlier than expected, and 118 or 16.7% discontinued treatment itself earlier than expected. In many of these, the outcomes can still be ascertained. For example, those who die or have late positive culture, they represent about 30% of those who, they represent about 25%, the deaths down here and the positive cultures here, of those who discontinued treatment earlier than expected. But in some of these other categories were constrained, like in withdrawal of consent. These others, lost to follow-up, investigators' judgment, were constrained in ascertaining an outcome. And that's something that we really want to avoid in the trial, in order to make sure that everybody's contribution to the trial, all these participants' contribution to the trial, isn't lost. Here, we're giving you the first snapshot of results in the NTB trial. These are aggregate. We're not showing you anything by arm. And these were data that we shared, or results that we shared with the Data Safety and Monitoring Board at our last meeting in July. And so these data have an earlier cutoff of May of 2021. In aggregate, at that time, 541 NTB participants had sufficient follow-up to be eligible for assessing their 8-week endpoint. That means that they had been in the study for at least 8 weeks since the randomization, plus time for culture results, plus time for data entry. And the exciting provisional news is that nearly 75% were culture negative at 8 weeks, while 14% were still positive. We can't ascertain culture positivity or negativity in the remainder, and just over 10% for the reasons shown. The withdrawal of consent and the sputum not collected are the most worrying, as we will never be able to change that. And again, we don't want to compromise the study results by missing data. Now moving to the 39-week outcome. So 440 were eligible for assessment of this outcome. In some cases, those outcomes had already been validated. In some, they hadn't yet been validated. So there might be modest changes. 83% had a favorable outcome at 39 weeks, and 21% had, it's about 5%, 21 individuals, about 5% had unfavorable outcomes, either microbiologically unfavorable or death. And then these others are also classified as unfavorable initially, but it's really more of an issue of not being able to ascertain them, in which case we have to classify them as unfavorable. We would like to avoid that again. Moving on to the 73-week outcome, which is the primary endpoint for the study, as you'll all remember. And also, so in this case, there were 355 who were had outcomes eligible for analysis, some validated, some not yet validated by the CAC. And almost 80% had favorable outcomes at this point. There were 7 deaths, and the same number with microbiologically unfavorable and slightly more clinically unfavorable outcomes. The rest we have challenges in ascertaining those outcomes, and they could be lost from an analysis population because of that. Also from the DSMB meeting is just a little snapshot of the safety data that you guys are so meticulously recording and reporting. This also This is also from May, and you can see that nearly all study participants had at least one adverse event. Half had an adverse event of grade 3 or higher. Nearly 15% had at least one serious adverse event, and sadly, in 10 participants, this led to death. 20% had the adverse events of special interest, and you can see that the hepatotoxicity and hematologic toxicity were the most common ones. As in output 1, grade 3 QTc prolongation has been much less common than feared. So far, that's encouraging for regimens containing bedaquiline and/or delamanid. Now looking at the summary of major deviations in these major categories, which is again the way we report them to the Data Safety and Monitoring Board and also discuss them with the DSMB. You can see that there were 179 deviations until May 15th, 2021. The most common are safety reporting delays and treatment administration errors. Just want to note that some of these treatment administration errors, for example, the use of prohibited concomitant medications, can result in having to exclude patients from the analysis. So again, in addition to ensuring the safety of the participants, we want to avoid these deviations that might have implications for the completeness of or confidence in our study results. I just want to provide a brief summary also of some of the protocol changes that are in the offing in version 3.5. You've heard about these before because this has been pending for quite some time in most of the countries. So far, version 3.5 of the endTB protocol has been approved only in Lesotho. But we're waiting in other countries. I am not going to go through those changes that are related to eligibility since we have completed enrollment, but I'll discuss those when we talk about NTBQ. Related to ongoing study participation, you can see that we have added a new weight band, a lower weight band for dosing guidance, and particularly this change in weight band has implications for bedaquiline, which is the dose for which is cut to 200 milligrams daily during the loading period, and then followed by 100 milligrams daily 3 times per week for its duration. Delamanid dosing is cut in half, and then linezolid dosing is also cut in half until week 16 and the timing of the dose reduction randomization. Some of the other changes, just wanted to mention that it includes the kind of validation of the practice of having home and virtual visits as necessary to protect staff and participants during the COVID-19 pandemic, the addition of amikacin drug susceptibility testing as a routine test, and adding sequencing as a type of genotyping allowed by the protocol. We've also clarified some information on the linezolid dose randomization. There's no change in it, just some clarification. And then there are a few sort of administrative changes that include removing or adding countries and sites as relevant, updating the study timeline to reflect where our information was at that time about completion, and then making some changes to some of the analysis specifications and populations. Now I'm going to transition to endTBQ. They'll be roughly the same set of slides with a few differences. Please note that we, as previously mentioned, celebrate the addition of Vietnam 1 in Hanoi to the endTBQ team. The site was activated in September of this year and is now screening. At extraction of these data, so this was at the end of October, I'm sorry, I see the date is missing, 101 participants, or 28.5% of the target enrollment, had been randomized. And while we remain behind our projections, we are catching up, and we project completed enrollment in January of 2023 with last patient last visit in mid-2024. The pace of enrollment and the timing of all this can be changed dramatically pending with the activation of additional sites in Mumbai, in Ho Chi Minh City in Vietnam, in Almaty, and in Nur Sultan in Kazakhstan. The Lesotho site did stop enrolling endTBQ at the same time that endTB enrollment stopped, just because of low numbers, unfortunately. Peru 1 and 2, which were anticipated to stop enrolling with endTB, had a little bit higher enrollment than we had been seeing, and so they're for now continuing provisionally through January 23rd. And if their contribution is steady and the costs are manageable, they may continue enrolling in endTBQ. We will evaluate that in January. Excuse me, sorry. This is the same disposition slide that you saw for endTB, but now for endTBQ. So 419 patients had been screened for endTBQ at the time these data were locked. You can see there's a much higher apparent exclusion proportion of 78%. Many of those who were excluded from Q went into endTB, and we're not showing that here. As with endTB, the most common reason for exclusion is related to some assessment of MTB or resistance on molecular tests. And we're looking to improve where we can. For example, we want to minimize the number of missing results where indeterminate results are a consequence of improved sputum samples. We'd like to try to where indeterminate results are a consequence of poor sputum samples and we can improve the samples, we would like to do that. And we'd also really be interested in exploring where the site study labs are with the introduction of expert MDR cartridges. And we will talk a little bit more about this in the exclusion section, but you can see that there were 8 individuals whose participation was excluded because of indeterminate results. Currently, Protocol 4.1 permits expansion of inclusion for inconclusive results on the MTBDR-SL test in Peru and Vietnam if the participants have also had prior exposure to fluoroquinolones. That is their inclusion is already permitted in India and Pakistan, where there are higher background levels of quinolone resistance and there's no prior exposure requirement. I want to highlight that 13.1% of the exclusions were due to investigator discretion. This is 3 times higher than the percent in endTB, and something that we watch closely. We really want to bring that number down, and we want to encourage investigators to limit their exclusions to being for protocol-defined reasons. And we find that often when when investigator discretion is selected, there is something related to one of the protocol-defined exclusion criteria. It just isn't elected. And one of the topics we might consider discussing when we're together is what you know about the use of the criterion in your site and why it's used frequently, if it is. And again, as mentioned before, the prior exposure or resistance to study drugs as a reason for exclusion remains low, and we'll just want to keep an eye on that. Just to give you a sense of the variability of exclusion by site, this red line shows the bar where you look where the exclusions are. If you look across all sites, and you can see just that a few of the sites have a higher rate of exclusion. I don't think we can really interpret anything from Vietnam, given that that's only 2 patients who have been screened and neither was eligible, unfortunately. But it's just worth keeping an eye on. And with PE2 being so low, one wonders if there's a lot of pre-screening happening that's just not getting documented as official screening. These are the reasons for exclusion when it gets broken down, and you can just see that in PE2, there's a substantial proportion due to investigator discretion, and a little bit of that showing up everywhere so far except Vietnam. So as I mentioned before, this would be something we'd really like to talk about when we're gathering. Gender ratio is very similar to that in endTB, with about 45% being female, and the population is still quite young. This is the comorbidity distribution. It's also similar to that observed in endTB, except for the prevalence of HIV being much lower in endTBQ. This is likely because South Africa is not participating in the endTBQ study, and only small numbers from Lesotho were included. Just take this opportunity to remind everyone that patients should not be excluded from the endTBQ study for any of these comorbidities. These are not inherently exclusion criteria. They may have other reasons to be excluded and also have these comorbidities, but the comorbidities themselves should not be a reason for an exclusion. And this is it's going to be even more important from the perspective of getting generalizable results to people living with quinolone-resistant TB and also with HIV. It will be even more important to include those in the remaining sites in endTBQ, since we don't have Lesotho in South Africa to help with that very important subpopulation. Same is true for Hep B and Hep C. You can see there's a slightly lower proportion of those with prior TB treatment and treatment on screening. It might be a difference of a function of the difference in those sites that are participating in endTBQ compared to endTB. I had previously mentioned Kazakhstan as kind of an outlier in the amount of prior treatment and treatment on screening, and Kazakhstan does not figure into endTBQ yet. The disposition of studies that have been randomized were much earlier in the process for Q, so it's a lot harder to say anything, but the signs for retention are good so far. Congratulations to everyone. There's only 1 patient who's discontinued follow-up prematurely to date. 3 unfortunately have died or had a positive culture at week 16 or later, and we'll keep watching these trends. For endTBQ, very, very provisional results that were shared with the DSMB back in July. You can see that there were only 18 endTB participants who had enough follow-up to be eligible for assessment of the 8-week culture result. And of those, more than 75% were culture negative, none were positive, and there were no missing results for this indicator, which is great news. On the safety for this population, there were 49 patients included, randomized at this point. And as with endTB, nearly all, more than 80%, had at least 1 adverse event. About a quarter had a grade 3 or higher, or an AESI. In this case, peripheral neuropathy was most common. Again, very little QT prolongation, only one case, of grade 3 QT prolongation. And I want to draw your attention to the fact also that there was no hepatotoxicity reported to date. We don't know yet why there would be such a big difference between endTB and endTBQ, but it could be related to the fact that pyrazinamide is included in all of the endTB experimental regimens, and it is not part of endTBQ. Just a reminder, 11.5% of endTB patients with data had an AESI, hepatotoxicity AESI. Deviations, similar, very early in small numbers. We can't really say anything, just note that there were several treatment administration error deviations. Now, back to the protocol changes, this time for version 4.1 of the endTBQ protocol. These have been pending a long time. We were waiting for a central IRB decision on the protocol, and it's now back in our hands, and we will be working on a few more changes, which we'll discuss with you on another occasion. They haven't been fully worked out yet. The big eligibility related changes are that there is no longer an exclusion based on low albumin, and also the threshold for exclusion based on QRS complex duration is now at greater than 120 milliseconds. As I mentioned previously, broadens the results that can be used from samples collected less than 3 weeks prior to enrollment in the study, and also there are certain other standards that have to be met by those samples. I note also that the undefined MTBDRSL results can be allowed in Peru, as I mentioned previously, and Vietnam, with evidence of prior quinolone exposure. This other exclusion criterion related to participation in another trial, we've made it more clear now that that participation in endTBQ is only precluded if somebody is participating in another trial that uses that is either testing TB treatment or testing a medication that is used in other cases for TB treatment. We have changed the contraception requirements to align with European guidance. So a double method is not always required, and some previously allowed methods may no longer count. We encourage you to review the SOP on this and ask your central study coordinator if you have any questions. We've also clarified the requirement that in the case where a witness is participating in informed consent, that witness must be able to read and write. Related to not eligibility, but ongoing participation, randomization, we have modified and clarified the extent of disease evaluation process. So while it's evaluated first at the time of randomization based on smear results and chest x-ray, and a decision is made at that point whether a participant is eligible for the 6-month regimen or the 9-month regimen. Only excuse me, only patients who had been assigned the 6-month regimen are eligible for reconsideration of the duration based on treatment response. So with culture results starting at week 8 and anything available before week 24, if there are positive results, then those participants will be shifted from the 6-month regimen to the 9-month regimen. It is not possible to shift the other way based on treatment response. So a patient who's been assigned to a 9-month regimen will remain on a 9-month regimen regardless of treatment response. Coming back to some of the other changes, the previously mentioned flexibility about COVID visits and the weight band, and inclusion of information on COVID are also in the endTB version 4.1 protocol. And there aren't really any other major changes that affect the trial operations at this point. We will update you on more possible changes moving forward. So I really just want to reiterate our deep congratulations and gratitude to all of you for your work that would have been amazing even without the COVID-19 pandemic, and that is nothing short of extraordinary in your adaptation and your continued commitment to participants in the pandemic. Just caution that even though I've shown you some provisional results, we can't say anything about these outcomes yet. There's no interpretation that can be made. And just a few reminders about the importance of including everyone who is eligible according to the study criteria and avoiding exclusion of patients for reasons that are not specified by the protocol. And avoiding discontinuing study treatment unless it's necessary for patient safety. Just noting that the prolonged QTc interval does not appear to be a major issue so far. Hepatotoxicity appears to be more important in endTB than in Q, but we'll have to wait and see what the additional accumulation of data from Q tells us. And just want to keep reinforcing the importance of completing all the study visits, collecting all the samples that permit outcome assignment, and supporting our participants all the way throughout their participation, and that mistakes are going to happen, and we just need to own up to them and learn from them. Oops. Sorry, going the wrong way. So these are my prompts for questions or discussion that you can include in our discussion platform, or when we convene to have a discussion about this talk. Happy for you to raise these or anything else. And I will leave it at that and look forward to being together with you all soon. Thank you.